Welcome to the Organic Chemistry Podcast, Dr. Brian Lloyd's scribblecast of organic chemistry lectures and solutions to homework problems. In this scribblecast lecture, we are beginning to look at amino acids. Alpha or two amino acids have a CO2H group and an NH2 group. So they possess both an acidic group and a basic group. Now, although there are approximately or over 500 different amino acids that actually occur in nature, only about 20 of amino acids are used by all organisms in the biosynthesis of proteins. The distinguishing feature between these amino acids is not the CO2H or the NH2. In fact, all amino acids possess those two functional groups. The 20 biologically important amino acids differ only in the R substituent side chain. Now, R often has just meant an alkyl group, but in this case it can mean any type of functional group off of this carbon. Most often there is a carbon, but it could be hydrogen, it could be any others. Now, they are not typical organic compounds in terms of the physical properties. Amino acids actually have melting points greater than 200 degrees Celsius. They tend to be soluble in water. So just like carbohydrates, they're different than most organic molecules in that they're soluble in H2O. In terms of acidity and basicity, amino acids are less acidic than most carboxylic acids and less basic than most amines. Typical carboxylic acids are C double bond OH. Can have quite a spread in pKa, but the pKa for a typical carboxylic acid is usually around 5, approximately. Okay. The pKa for an amino acid are CH, CO2H. Let's put the NH2 up. The typical pKa for amino acid is approximately 10. Now, amines, you measure base strength in terms of pKb, so let's write pKb down here. Amines usually have a pKb around 4. The pKb for amino acids is around 12. So, a question arises. How can we explain this difference in pKa and pKb from free carboxylic acid or free amine when compared with that of the amino acid? Well, the answer lies in close inspection upon the chemical behavior that we would expect for an amino acid. The carboxylic acid group is an acid group, which means it donates protons. The lone pair on the NH2 is a basic group, which means it accepts protons. Hence, if we dissolved an amino acid in water, we would expect an equilibrium with the following structure. Where the CO2H has lost its proton, and the lone pair on the nitrogen has picked up the proton. In other words, in aqueous solution, the carboxylic acid would protonate the NH2. Now, which side of this equilibrium has the greatest concentration would depend on the acidity. That is, the CO2H, is it a stronger acid than the NH3+? See, NH3+ is the conjugate acid of the base NH2. So who's stronger, the acid or the conjugate acid? Indeed, NH3+ is a weaker acid than CO2H. Hence, as I've drawn, the equilibrium shifts to right. So when you take an aqueous solution of amino acid, you're actually measuring the acidity of NH3+. You're measuring its acidity. And since it's a weaker acid, no wonder the pKa does not match that of a carboxylic acid. Likewise, CO2- is a weaker base than NH2, which has a lone pair. CO2- being a weaker base, when you measure pKb of an amino acid, you're measuring the pKb of CO2-, not NH2. Hence, the basicity, or pKb, is less than we would expect for a base like NH2 or an amine. The ability for this type of ionic species to form also probably contributes to the observed physical properties of higher boiling points and the molecule's increased solubility in water. Excuse me, I should have said higher melting points, greater than 200 degrees Celsius. All right, let's discuss stereochemistry. Stereochemistry of amino acids. As you can see in the structures I've drawn, there is a clear chiral center in this molecule. As a result, we can draw Fischer projections. Now, if you remember in carbohydrates, the standard Fischer projection was based on the molecule glyceraldehyde. And this molecule, L-glyceraldehyde, was a mirror image of D-glyceraldehyde and served as the precursor to the nomenclature of all other sugars. Well, in amino acid chemistry, we draw the molecule much the same way in the Fischer projection. We have CO2H. We put that at the top. we put the R group at the bottom. And then we have the NH2 and the H. Now, if the NH2 group is to the left, we have an L-amino acid. And because of this relationship with diglyceraldehyde, it's adopting a stereochemical structure similar to diglyceraldehyde, and this is the way we apply the L. The mirror image of this, of course, is the D-amino acid. Always remember that in a Fisher projection, the horizontal line is coming out at you. The vertical line is going away from you. Now, don't forget that. In other words, if I was to put the central carbon in, we'd have dashes in the vertical line, meaning they go away from you, and wedges in the horizontal line, meaning those two groups are coming out at you. So that's the standard format for a Fisher projection. That is what the intersection of the two lines really means. Okay. Now, D-amino acids are found in some bacteria and fungi, but the most predominant proteins have the stereochemistry all in the L configuration. There is one exception to this. This was published in November 15th, 1999 in Chemical Engineering News. It simply states that D-serine, one of the amino acids we're going to look at, is actually synthesized in mammals' brains and acts as a neurotransmitter. D-amino acids are generally thought not to occur in mammals, although they are prevalent in bacteria. Recently, however, a few researchers have reported finding high levels of D-serine in mammalian brains and have shown that in concert with glutamate, it activates the N-methyl-D-aspartate NMDA receptor. Activation of this receptor has been linked to memory and learning, among other brain processes, as well as to nerve cell death and stroke. The source of mammalian D-serine has been a mystery. Its origin variously described to diet or intestinal bacteria. But these individuals have shown that D-serine is synthesized in the brain from its enantiomer L-serine. The team cloned and expressed serine racemase, the enzyme that performs the trick, from mouse brain extracts. The researchers stated it may also have important far-reaching implications for thinking about the function of D-amino acids in mammalian biology. Okay? So, there you have it. Although we typically work with D-amino acids, typically work with L-amino acids in biological systems, there are examples, although they are rare, of D-amino acids that are important. And their discovery is certainly going to probably continue in the future. So what we want to do is now take a look at these 20 major biologically important amino acids. Okay, so these are amino acids used by all organisms in protein synthesis. Okay, and the amino acids which are required for protein synthesis and are broken down into two categories: those that we can synthesize and those that we can't synthesize, hence they must be consumed in the diet. Essential amino acids, by definition, essential amino acids are those amino acids that cannot be synthesized. and hence must be present in the diet. So, I'm going to asterisk the essential amino acids. See if I can do that a little better. So we're going to look at amino acids individually, one at a time. Look at the names, abbreviations, and indicate whether they can be synthesized or whether they must be consumed. The first one, and we're going to do it alphabetically, is the molecule alanine, and you are responsible for all of these names, structures, and abbreviations. Alanine. Given the abbreviation ALA, and you are responsible for the abbreviation, this molecule can be synthesized biologically. And we're going to just draw them not in the ionic form. We'll just draw them in the standard amino acid form. So I will put the R groups pointing down. So here we go. Here's your standard amino acid group. What does alanine have as the R group sticking off of it? Alanine has a methyl. A methyl. All right. Now, one of the things we're going to look at later is what type of amino acid this is. Really, to answer this question, we're going to need more information. So we're going to come back to this. We'll review these amino acids once we learn the distinguishing feature between the types. Now, type is whether or not the amino acid is an acidic amino acid, a neutral amino acid, or a basic amino acid. And so there is this distinguishing feature between the amino acids, whether you're acidic, basic, or neutral. And we will look at that later on. We'll be coming back to that. Another amino acid is arginine. Arginine. Now, arginine has the abbreviation ARG. And this one is an essential amino acid, okay? So, you need to know that. Now, if I continue drawing them the same way, I have an H2N, a CH, a CO2H. What is the R group for arginine? It's a rather complicated fragment. It has 3 CH2s, and then off of the last CH2 is an NH attached to a C. Off of the C is a double bond, NH, and off of the C on the other side is an NH2. There you go. And just so you can know, there's a lone pair here. The NH with the double bond has a lone pair, and the NH2 has a lone pair. So there's a lot of lone pairs off this end. Okay. Asparagine. Or asparagine. It's given the abbreviation ASN. You might wonder why it's given ASN, but that'll become clear once you see the next one, because the next one is called aspartic acid. Hence, aspartic acid, as you might guess, is given ASP. And you could not give two different amino acids the same abbreviation. It would be confusing, so they needed one and they chose N for asparagine. Now, asparagine If I draw these the same way, and you may notice it drawn differently in your handout notes, but you should be comfortable with seeing it drawn different ways. So here's asparagine, and asparagine has a CH2, then a C double bond O, and a asparagine and NH2. So it has an amide functional group here. So let's draw some lines. That might be good to split these up. So it's a lot clearer which name goes with which structure. There we go. Well, what about aspartic acid? Well, instead of the amide unit, aspartic acid is very related, as you might guess. A similarity in name suggests there's a relationship with asparagine. Indeed, structurally, they are similar. You have a CH2. Okay, and you have a C double bond O. I'll have to draw it up because I'm running out of room. But instead of the NH2, you have an OH. So aspartic acid has a carboxylic acid off the R group, whereas asparagine has an amide functional group. Only arginine in this list of 4 amino acids. Only arginine is an essential amino acid. Let's move on. Taking a look now at the amino acid cysteine. Cysteine I mentioned previously in the alcohol last semester. Alcohol section, of course, actually in the section of thiols, being responsible for curly hair, and cysteine was mentioned as an amino acid. Then CYS is the abbreviation. Sometimes the S is given as a capital S. Either is acceptable in this course. You have the structure NH2, okay, H2N, attached to a CH, and our CO2H, a standard carboxylic acid framework. And to make a cysteine, I said thiol, we have a CH2 with a thiol group SH off of it. We've got glutamic acid. Glutamic acid has the abbreviation GLU. And glutamic acid is very similar to aspartic acid. So memorizing it's not bad. You just have to keep straight the number of carbons. Aspartic acid had 1 CH2 and then the carboxylic acid. Glutamic acid, it's 2. And just like aspartic acid had the related amide asparagine, glutamic acid has the related amide glutamine. Again, we could not use GLU twice, so like asparagine, we use GLN as the abbreviation. And so, similar to glutamine, similar to asparagine, glutamine has a carbon chain before you hit an amide group. Whereas asparagine had 1 CH2, glutamine has 2. And so these are very related. Okay. And instead of a carboxylic acid, such as in glutamic acid, we have an NH2, which gives us an amide functional group. Glycine is an interesting amino acid. GLY is the abbreviation. It holds the honor of being the only amino acid that lacks what? A chiral center. It has a hydrogen off the carbon. And because there's two hydrogens, the center is no longer chiral, so glycine is not chiral. Histidine is a very interesting molecule. We're going to look in more detail and explain what I'm about to, the information I'm about to give you. Histidine is a molecule without a specific structure. What? What do you mean? Well, in actual fact, it has a specific structure, but you'll see what I mean in a moment. HIS is its abbreviation, and I'm going to asterisk it because it is an essential amino acid, which means it must be consumed. Histidine, how can I draw this? Has a ring structure as the R group. What's amazing about this is where that ring structure attaches. It's a 5-membered ring with 2 nitrogens in the 5-membered ring. So it's a lone pair here, and 2 pi bonds. Interesting. But it doesn't just have this structure. It also can be drawn in this fashion. With the point of attachment of the amino acid in a different position. Now I'm going to give you a clue and leave it for a little later on in the course. We're actually going to do some questions later on that will help explain this. So jump ahead actually quite a bit closer. When we start talking about types of amino acids, we're going to look at histidine again, and it will become clear why it has these 2 structures. The only thing I'm going to say about it right now is that at the time, we will see that these 2 structures are actually in equilibrium with each other, and they are tautomers. Although it doesn't look like it. Remember what tautomers were? Tautomers, remember, are structures in which you have shifted pi bonds and 1 or more hydrogens. And if you look carefully, in reality, that is what we've done. If you take this structure and move, I'll circle it, this proton to that nitrogen, okay, and then shift this pi bond here. Do some shifting of pi bonds. you would actually get, okay, so we move these pi bonds around, you would actually get this structure. It would be. In any case, we'll look at this more closely a little later on. They are related to each other as being tautomers. Great. Moving right along. After histidine comes isoleucine. Isoleucine is another essential amino acid that must be consumed in our diets. And if we draw structures, H2N, isoleucine has an alkyl group attached to it. Can you give me a common name for this alkyl group? What is this alkyl group called? It's a sec-butyl. The R group is a sec-butyl group. You have isoleucine. Leucine, instead of a sec-butyl, it's also an essential amino acid. Has an isobutyl group. So hopefully you remember your alkyl substituents from last semester. Lysine. Lysine has the abbreviation LYS. It is an essential amino acid. And lysine has a very large chain for its R group. It consists of 4 CH2s. Four CH2s and then an NH2. 4 CH2s and an NH2. Lysine. Methionine is probably most closely related to cysteine. It's also an essential amino acid. It's given the abbreviation MET. And I say most closely related to cysteine. It doesn't have one CH2, it has 2 CH2s, but it has a sulfur in it. It's not a thiol, but it's more like a thioether. So there's a sulfur followed by a methyl. The next one is phenylalanine. And as the name suggests, this is simply a phenyl attached to the alanine. If you remember alanine had a methyl as the R group, so if we attach a phenyl to that methyl, it must become a CH2. It's given the abbreviation PHE. Phenylalanine is an essential amino acid. So again, we have our basic framework for amino acids. And remember in alanine, the substituent was a methyl. Well, in this case, it's a CH2 because there's something attached to the methyl. It becomes a CH2. And what's attached to it? It's a phenyl group, just as the name suggests. Okay. So there is phenylalanine. Proline is a unique amino acid because it lacks the typical framework. In fact, the standard framework of NH2CHCO2H is there, but the CH is part of a 5-membered ring, and you don't have an NH2. What do you mean? This is proline. Now, if I was to draw a circle around the fragment, the standard fragment for amino acids, that would be this fragment. So you see we have a CO2H, a carbon with a hydrogen, but instead of an NH2, we have an NH that's in a ring. That makes our R group side chain these 3 CH2s that form the rest of the ring. So it's a bit unusual for that reason, and probably takes some memory work to learn. The prefix, of course, for proline, or the abbreviation for proline, is pro. Proline is not an essential amino acid. The next molecule we've already talked about was serine. This was the one that L-serine is converted to D-serine in mammalian brains. Serine is not an essential amino acid, has the abbreviation SER. And we've got our basic framework. It's very closely related to cysteine, except instead of an SH, we have an OH. CH2OH instead of a CH2SH. Next is threonine. Which is an essential amino acid. Threonine, given the abbreviation THR. Again, our basic framework, H2N, CH, CO2H. Off of that basic framework for threonine is a CH with a CH3, very related to serine, except the CH2OH, the CH2 unit, has an extra methyl placed off of it. Okay, so the OH is off this carbon, right off the fragment. Tryptophan is another molecule with a large side chain. Tryptophan. Another essential amino acid, so let's asterisk it. And it's given the abbreviation TRP. Drawing our basic framework. Notice I am not drawing Fisher projections here. That's pretty obvious. Well, what do we have? Well, what we have here I may have troubles drawing this. Hang on. In fact, let's try drawing it this way. That's what I'll do. Okay, so this has an NH here. And a 6-membered ring. And a double bond here, and the attachments right here. Okay, so this is the molecule tryptophan, which is essential amino acid, and I did forget something, sorry. There is a CH2 between the ring structure and the amino acid end. There we go. Okay. Next we have amino acid called tyrosine. Tyrosine is not essential. Remember, essential does not mean required. It just means you must consume it. Okay? Non-essential amino acids are also required. In other words, all these amino acids are required. So don't just think essential amino acids means it's essential in the sense of being required and the others aren't. They're all required. It's just essential amino acids can't be synthesized, so you have to have them in your diet. Okay, tyrosine has the same fragment. And directly bound, we've got a CH2. Tyrosine looks a lot like phenylalanine. In that, you have an aromatic ring attached to a CH2. By the way, it's very easy to forget that CH2, so be careful. But para to CH2 is an OH group. So tyrosine puts an OH para to the CH2 group. Excellent. Okay, last but not least is the amino acid valine. Valine. Now, valine, given the abbreviation VAL, is an essential amino acid, so we better asterisk it. If I draw my amino acid framework, and draw valine, valine is probably more closely related to leucine and isoleucine, and alanine. It has an alkyl group off of it, and the alkyl group is this group, which is called, what's that group called? Isopropyl. So valine has the isopropyl group off of it. Now, you'll notice I have listed a lot of amino acids as essential and a lot as non-essential. The non-essential simply mean they can be synthesized. Most non-essential amino acids are synthesized in what is called the Transamination reaction. The transamination reaction. Okay. Now, in the transamination reaction, there is a transfer of an amino group from an unneeded amino acid to a keto acid. Now, I'll give you an example, and you are responsible for knowing this example. So, suppose I had the amino acid alanine. And I reacted with acid referred to as alpha-ketogluteric acid, an acid found in biological systems. Alpha-keto, the carboxylic acid's located here, the alpha carbon is here, so if it's alpha-keto, that means it's a ketone. And glutaric acid, remember, had two CH2s. And a CO2H. Essentially, what happens here is through a process involving an enzyme, okay, and I'm going to name the enzyme transaminase. Which is an enzyme. This H and this NH2 end up going where the C double bond O is. And the C double bond O ends up going where the H and NH2 was. Now if that happens, you're going to end up with CH3, C double bond O, CO2H, which is pyruvic acid. And you will have CO2H, C, okay, NH2, H, CH2, CH2, CO2H, which is glutamic acid. So with the enzyme, transaminase, one amino acid can be reacted with these alpha-keto carboxylic acids to produce new amino acids. Now, this conversion of this group, in fact, it is the keto group, okay, that will react with the transaminase after the transaminase is reacted with the amine. Essentially, the transaminase makes an imine with the amine group, and then you get a conversion with the keto acid in the final step to produce the new amino acid. Now, we will be looking at these types of reactions when we look at aldehydes and ketones and imine chemistry. So there is an enzyme-bound aldehyde group which is involved in this reaction or this conversion. So I expect you to know this as an example for how non-essential amino acids can be synthesized. All right. The last thing I want to cover in this Scribblecast lecture is that our discussion of what is essential amino acid does not always apply to everyone and what is non-essential. For example, what we want to do is look at a genetic disorder called Phenylketonuria. Phenylketonuria. Tyrosine is not considered an essential amino acid because most people can synthesize it from phenylalanine. Okay? In other words, most people can take the amino acid phenylalanine and they have this enzyme called phenylalanine hydroxylase. Hydroxylase. And once they do this reaction, they actually produce an OH. And they make the molecule tyrosine from phenylalanine. However, some people suffer from phenylketonuria, given the short name version PKU. Now, this is a genetic disorder in which a person lacks the enzyme phenylalanine hydroxylase. Because of this, they cannot convert phenylalanine to tyrosine. Since the conversion cannot take place, the amount of phenylalanine builds to excessive amounts and takes the alternative pathway to phenylpyruvic acid. So in the person who suffers PKU, or in the PKU victim, we get pyruvic acid, phenylpyruvic acid, which means HO2C. Instead of having an H and an NH2, we get a double bond O here. And you don't get the OH on the ring. Now, the diet of these such individuals must therefore have tyrosine. For most people, tyrosine is not an essential amino acid because they have the enzyme phenylalanine hydroxylase. But for a small percentage of the population who have this genetic disorder, because they lack the enzyme, tyrosine becomes an essential amino acid. So their diet must have some tyrosine, and because of the buildup of phenylpyruvic acid, their diet must be limited in the amounts of phenylalanine. Okay. Excessive amounts of phenylpyruvic acid, if it builds up soon after birth, can cause severe mental retardation. So it's very important that this be identified at an early stage. And so the tests are often done on newborns to determine if this is the case. PKU is sometimes given the term phenylketones in the urine. And so they're looking for the excessive amounts of phenylpyruvic acid. So the reason I want you to learn about this genetic disorder is to indicate while there are some standard norms for individuals, there are variations within populations that one amino acids that's non-essential because of a genetic disorder can become essential. And another essential amino acid, such as phenylalanine, in those cases, you may have to actually watch your consumption of these amino acids because of the harmful side effects. All right. Well, I'm Dr. Brian Lloyd. This is my Scribblecast, and we have just begun looking at amino acid chemistry. Thank you for listening.